Thank you for the introduction, and I would like to thank the organizers for the invitation to speak here. So honestly, my main theme is not large deviation theory. So I'll tell you this story and show you how at the end we also use a tool from a large deviation theory to do calculations here in this system. Okay, so biological systems often need to evolve under multiple and possibly conflicting selection pressures. And the question that has fascinated me for a long time is how can they reconcile such conflicting selection pressures? And today I will discuss this more general question with you in the context of a particular system in plants that my lab has been working on for the past few years, and that is the self-incompatibility system of plants. So first, since there are no plant biologists here, what is self-incompatibility in a nutshell? So many plants are bisexual. That means that they have both male and female organs within the same flower. So this is the male organ, the anther. This is the female organ. These are the ovules here at the bottom in the ovary. And simply because of the physical proximity between the male and the female organs within the same flower, then a large proportion of the pollen that arrives here is coming from the very same flower and not from other plants. Okay, and once the pollen arrives here, then it should go all the way down to the ovary. It can fertilize, form offspring. And if this is pollen coming from the same flower, then this is self-fertilization. And the problem with that is that the large proportion of the offspring could suffer from genetic defects or even be inviable. So in order to reduce or even eliminate self-fertilization, plants developed various mechanisms. Some of them are called self-incompatibility, and this is what I'll be talking about. So self-incompatibility means that the species is subdivided into multiple distinct types, typically a few dozen of them, such that fertilization is only possible between male and female gametes that have a different type. So here in this schematic example, the plant that is receiving the pollen has type S1, S2. So these are the ovules here at the bottom. And pollen that has the same type, either S1 or S2, is simply blocked by this mechanism and cannot fertilize. And only pollen that has a different type, here it's S3 or S4, can go through, reach the ovules, fertilize, form offspring. So how is that determined? So the type that I refer to is encoded in a particular genomic locus of the plant called the S locus or S haplotype. It encodes several genes that are expressed either in the male or in the female reproductive organs. And different types have different alleles of these genes in this S haplotype locus. And as I'll show you, they implement some sort of molecular recognition to identify what is the type of the pollen that is coming. Yeah, sure. At least 20, highest I heard of is 60. These are the orders. By the way, for different plant families, also there is more than one mechanism like this. At least 3 were described, different molecular implementations, and the numbers are very similar in all of them. I'll be talking about just one of the mechanisms. Okay, so let's say, so I'm focusing here on one particular mechanism. Okay, there are several others. Let's take a closer look at the genetic architecture of this S haplotype. So it contains a single gene that is expressed in the female organ. It is the protein that it encodes for is toxic and for the pollen, and by default inhibits fertilization. For people more interested in the biology, I can say a bit more on that later. And the others are genes that are expressed in the male organs, and they encode for proteins that can specifically recognize some of the female proteins and then target them for degradation and by that enable fertilization. So as you see here, So each of them can specifically recognize one or several of the female types, such that collectively they should aim to recognize all the other types in the population except for the self type. Okay, so in this example, the female has type S1, and all the males collectively recognize types S2 till S6, but importantly, they must not recognize S1, because if they did, then self-fertilization would be possible. So another conclusion from this is that the functionality of the system depends on the combination of genes, okay? Because if you get here a male protein that would recognize the S1, then the mechanism would break down. So indeed, the recombination is nearly suppressed in this genomic region, and we will treat the entire combination as a single evolutionary unit that is co-inherited and only evolves via mutations. Okay, so what are the selection pressures under which these proteins need to evolve? So this is a very schematic description here. Imagine that this is some sequence space, okay, that I'll say a bit more next. And on the left, we have the sequences of these male proteins in a particular S haplotype, okay? So these are the red circles here. And on the right side, we have all the female proteins in the entire population. Every type here is a different color. And the self one is the red square. And the regions that I shaded here are the sequences that each of them can recognize and then target for degradation, enable fertilization, etc. Okay, so collectively, these three male proteins recognize all the female types in the population, but they avoid matching the self protein, the red square here. So the two types of selection pressures that we have here is, on one hand, avoidance of self-compatibility. So that means avoidance of the red square, the self-female protein. But on the other hand, promotion of cross-compatibility. So they should aim to match as many as possible of the other non-self types in order to find the mating partners. And what we'll study here is how do they evolve under such combination of selection pressures, just a second, when they could be possibly conflicting, because one of them means be more attractive and the other means be more repulsive. Yes. Okay, so one protein is in RNAs, okay? So the domain that it has a hypervariable domain, either 1 or 2, depending on the family, which is supposed to determine the specificity. The male protein is an F-box protein, which is part of the ubiquitination chain. Okay, so to answer this question, then we need a model that should account for the molecular recognition between these proteins. And we rely here on empirical studies that show that the specificity of these proteins depends on very few amino acids. So we represent here every protein as a sequence of L amino acids, which you can imagine as some binding interface, the binding domain of these proteins in which they recognize each other. And then we consider the molecular recognition here as an energetic interaction between a pair of one male and one female protein, and the total energy is the sum of the pairwise interaction energies between their amino acids. We also use here a simplified model where we consider only 4 biochemical categories of amino acids, either hydrophobic, polar, positively or negatively charged. We follow here, we borrow the model that is used in studying protein folding. And then if this total interaction energy between these two proteins is below some threshold energy that we set in the model, then these proteins are considered matching, and that means that fertilization between the S haplotype carrying the male protein and the other S haplotype carrying the female protein is possible. Okay, and if the E threshold, if the energy is above E threshold, then they are not matching. Fertilization is not possible. By the way, importantly, so every S haplotype serves here in both roles, both male and female, okay, depending on the context. So what is the meaning of this E threshold? That's a parameter that we set in the model. You can think of it as setting the promiscuity of interactions between these proteins. How likely are random sequences to match each other? So I'd like you to pay attention to several consequences of this kind of model construction. So first, every protein can match multiple different partners, because matching just means that the interaction energy is below the threshold. That means that the model has many internal degrees of freedom, because the same compatibility phenotype can be realized by multiple different genotypes. And another thing is fitness here is not something that is put by hand, as is often done in evolutionary models, but it's actually an emergent property of the sequence, which is its ability to find the mating partners in the population based on its matches and mismatches with other members of the population. So it's not a function of the particular sequence, it's actually a function of the entire population. Okay, and then we use stochastic simulations in which we study the evolution of a population of such entities containing, so the model was done in the protein level, not in the gene level, such proteins that evolve and constructed like this. So I'll show this. This is a description of the life cycle of the simulation. We have here a population of n such S haplotypes. Each of them is constructed of a single female and multiple male proteins, where every protein is such a sequence of amino acids, as I showed in the previous slide. And they go through rounds of mutation, in which potentially an amino acid can be substituted with some probability, and then reproduction. So ignore the left branch for the moment. So reproduction means that we pick at random parents to form offspring. So we pick a maternal S haplotype in the role of the female, and then we try to find the male partner. We just pick a random S haplotype and see if it matches or not, and if it does, then they form an offspring. So another important thing here is that by allowing multiple fertilization attempts here, we actually represent here asymmetry in the abundance of male and female gametes, which is actually the situation in nature. And that means that the female is almost guaranteed to be fertilized, okay, because we allow it multiple attempts to find the partner, and the competition on matching is mostly borne by the males, okay, competing between one another on finding compatible females. Okay, an important, yeah. So recombination could do different things, okay? So we're actually studying this now separately, so I can also tell. Recombination can do different things. The most harmful thing it can do is that introduce a male gene that recognizes the female gene, and then the haplotype becomes self-incompatible. It's able of self-fertilization, and then indeed some of the progeny will be inviable. So that's I think the most harmful consequence of this. So recombination is known to be nearly suppressed in this genomic locus. So these are very rare things that happen. Okay, this was shown experimentally. So this is, by the way, known from sex chromosomes. X chromosomes also have a low recombination rate. So recombination is non-uniform in the genome, and here it is suppressed almost. Okay, so one interesting result of this model is that following such a simulation of this population with exactly this life cycle over many generations, what we get is that the population spontaneously organizes into distinct mating classes or types, very similar to the mating types that we know from nature. And a class is defined such that within the class, every individual is incompatible with all the other members of the class. but each of them is compatible with all members of all the other classes, and classes are, of course, non-overlapping. And interestingly, almost all the population is classified into any of these classes. Okay, so that's something that we published, and I can talk a lot about that, but now I want to move forward to studying what are the underlying selection pressures that enable such an organization of the population. Okay, so we have here sequences that are in a setup of a large population, and each of them should both match and mismatch many other sequences in the population. And then we took these simulation results and wanted to analyze them to find out how do they do this. And before I show the results, and generally speaking, we thought of two main strategies in which a sequence can match or mismatch, which we call the global and the local strategies. I mean, you can call them also broad and narrow. I think something similar is known from the immune system. So global means that the sequence is on average attractive or repulsive towards an average partner or anti-partner. And that is done by just biasing its amino acid content in a position-independent manner, because it doesn't aim towards a particular partner sequence. And in order to see the impact of such a strategy, what we have to look at is just the marginal distribution of the amino acid frequencies in the protein and see if it is biased, okay, relative to neutrality. The other one means local, we call local. That means that the sequence evolves to either match or mismatch a particular partner or anti-partner sequence by specifically adjusting its amino acids towards those of the partner. And this is, of course, a position-dependent adjustment. And naturally, it can be done only for a relatively small number of partners. And in order to view this, what we have to look at is the joint distribution of the two partner sequences, because it, as I'll show you, it usually doesn't have an effect on the marginal distribution. Okay, so in general, okay, if there is no, so the thing that I will show you from now on is the signatures of the distributions of these amino acids. And if there is no selection, what we expect to see is that they follow the prior distribution, which is the distribution from which we pick the mutations whenever they occur. Okay, and whenever we see a deviation from this prior, which is the neutral state, we consider this as a signature of a selection acting here on these sequences. And I will use here the Kullback-Leibler divergence from information theory to quantify the extent of deviation. Okay, so this is a measure of divergence between the distributions P and the Q. Okay, so the first thing we wanted to look at is to what extent do the sequences that were evolved in our simulation deviate from the neutral one. So we did a very simple test. We took pairs, arbitrary pairs of male and female proteins from the evolved sequences and tested what is the probability that they match each other. And for reference, we also looked at the neutral sequences, just sequences drawn at random from the prior. And I'm showing this as a function of the energy threshold. Okay, so below these thresholds, sequences are considered matching. So for the neutral ones, it's very simple. The higher E threshold, then the higher is the probability that they match, of course. And as you see, we simulated almost the entire range from a 0 to 1 probability of match. For the evolved sequences, this is the red curve, we see something very different. So first, in most of the range, what we see is that the evolved sequences match much less than the random ones. Okay, so that means that the overall the net selection pressure acting on them is to avoid match rather than to increase match, which is a bit counterintuitive, because we normally think of the default of proteins is that they do not match, and then you have to apply selection to make them match. But here it seems to be the opposite. And it is actually in line with a very interesting experiment that people have done. that showed something like that in this system. So if I have more time, I can talk about it a bit more. And then we wanted to see, okay, so these sequences evolve to avoid match. So how is that done? So is it done by the global strategy, namely just biasing the amino acid content, or by something else? So the first thing is we looked to see if just a simple bias in the amino acid frequencies can explain this gap that we see between the neutral and the evolved sequences. So to test that, what we had to do is draw test sequences from the marginal distribution of the evolved sequences and see if they reproduce the same effect. So this is the gray curve here, okay? And you see that at least here at the high end, it can explain most of the gap between the neutral and the evolved sequences. That means that what we get here is mostly an effect of just a bias in the amino acid frequencies, making these sequences less attractive, okay? So this is the global strategy, but we still wanted to see what explains the remaining gap here. And here on the low end of the E threshold, then the global strategy is not explaining the trend that we see. So then we wanted to see if then the local strategy can explain the rest. Okay, so to find the effect of the local strategy, I'm reminding you that we need to look at the joint distribution of the two partners or anti-partners. And then we compared it to the product of their marginal. So whenever there is an adjustment, what we should see is that the joint distribution is different from the pair of the marginals because they evolve to be statistically dependent. Okay, so this is what I'm showing here. So it's the Kullback-Leibler divergence between the joint and the product of the marginals. And here we looked separately at the pairs that should mismatch, namely pairs that are on the same S haplotype and pairs that match. So these are partners on different S haplotype. This is the blue curve. So you see here that the local strategy has a major contribution to partner matching, okay? So here we get a deviation, significant deviation from 0. But only a very minor contribution to anti-partner mismatching. Okay, so that means that coming back to the previous graph, then most of the repulsion is done by the global and very little by the local, and most of the attraction is done by the local strategy. So another interesting thing that we found here, so this is coming back to the single amino acid distribution, the single protein, the global strategy. We found an asymmetric response to selection, okay? So here we're looking separately at the biases of the female protein in blue and the male protein relative to the prior. So we see that the female is the one that significantly deviates from the prior, where the male proteins remain almost neutral. And this is a bit unintuitive at the first glance, because I told you earlier that there is an asymmetry and the females are almost guaranteed to be fertilized, and the competition is mostly between males. So you would have expected maybe that those that should be biased are the males that are under pressure to match and not the females. Okay, but there is an explanation to this. And that will bring me to the large deviation part of the talk. Okay, so the last thing I'd like to show you is coming back to large deviation theory, how we use it to approximate the amino acid frequencies that we get here in this model. So first, I'm just reminding you that we saw that the net selection pressure is to mismatch rather than to match, okay, on average, okay, on all the sequences. So then we said, okay, why not take a simpler model, okay? So we took here a reduced version of the model in which there is only selection to avoid match within the haplotype, namely selection against self-compatibility, and there is no selection to match a non-self haplotype. So this seems like a very naive model, but quite surprisingly, it produces results that are quite close to the full model. And we tested this both in simulation, and then we analytically approximated the frequencies that we obtained in this simpler model when there is only selection to avoid a match within the S haplotype. So we use here a theorem from large deviation theory called Sanov's theorem, which gives us a bound on the probability of getting an atypical sequence from a given probability distribution. So in this case, the distribution from which we draw the sequences is the prior distribution, and we're asking what is the probability of getting such sequences that obey the constraint of no match within the S haplotype. And this theorem gives us the answer of what is the probability of getting such a sequence from gamma is the set of all the probabilities that obey this no self-compatibility constraint. And the probability is measured by the Kuhlblatt-Leibler divergence between this probability Q and the prior probability nu. Namely, in other words, what this tells us is that we will get the distribution that minimizes the Kuhlblatt-Leibler divergence relative to the prior from all those probabilities that obey the constraint that we imposed here. Okay, so the energetic constraint of no self-compatibility can be translated to some energetic condition. So that means that this is the interaction energy between sequences that are drawn from the distribution that we are looking for. And the condition is that the total interaction energy is above the threshold that we imposed here. And we are looking for distributions. Psi is the marginal distribution of the female, and phi is the joint distribution of the male and the female, such that they obey this constraint. And then we want to minimize their Kuhlblatt-Leibler divergence relative to the prior, and then playing a bit with the expression, we can translate this to pair distributions. And what we have to do now to calculate it is numerically minimize this expression subject to this constraint. Okay, so coming back to the symmetry between male and female proteins. So the first part here concerns only with the female protein, and the second part concerns with the joint distribution of both male and female. So you see here that there is a prefactor, which is the number of male genes. So the contribution of the male genes is m times larger than that of the female. So that explains why if you want to minimize the divergence, it is cheaper to only bias the single female protein rather than biasing the m male proteins, which explains the asymmetry that we saw. Okay, so these are results from both our simulations and the analytical approximation, showing here, in this case, the frequency of the hydrophobic amino acids as a function of E threshold. And I'm showing here, so in dark blue, it's the full model simulation. In light blue, it's the reduced model with only selection against self-compatibility. And the red one is the analytical approximation that we calculated numerically, and it should approximate the reduced model. And you see they are quite close to one another. And anyway, the prediction here is that these are the frequencies in the female protein. The prediction is that we get depletion of hydrophobic amino acids, and we also get enrichment of charged amino acids in the female protein. And we also looked at the genomic data of these female proteins, and we there too found a similar trend with the depletion of the hydrophobic amino acids in the domain that is considered to determine its specificity. And this is the prior, okay, this dashed line, the neutral, okay, so they are all below the prior. Okay, so to summarize, what I've shown you here is a toy model for the molecular recognition in the self-incompatibility system in plants. The model has many internal degrees of freedom and shows a degenerate mapping between genotype to phenotype. These proteins need to reconcile different selection pressures, both to match and mismatch other sequences in a setup of a large number of sequences. They use two basic strategies to do so, the global and the local. We find asymmetric response to selection between the male and the female proteins, and we also derive an analytical approximation for their frequencies under this simpler model. And we find that there is enrichment of the charged amino acids and the depletion of the hydrophobic ones, and this is in agreement with what is known from genomic data. This is the link to the manuscript. These are the people who did the work. So it's mostly the work of Amit, a postdoc in my lab, and it was started by Keren, who already left the lab. And it is in collaboration with Ohad Noi-Feldheim from the math department in the Hebrew University. And thanks for funding, of course, and thank you for listening.